I am going to be discussing my UI443 proposal for research. I am proposing to do a cyclic voltammetry study and determine the relationship between electrochemical behavior and biological activity of Quinoxalino 2,3-C cinnolines and their 5N oxides. Quinoxalines have been of great interest because of their wide range of biological properties and pharmacological uses. New heterocyclic di-N oxides are being synthesized every single year because of their biological properties, and they're being studied. And heterocyclic di-N oxides have been reported to have anti-cancer properties, antibacterial, antimycobacterial, antioxidant, anti-inflammatory, anti-trypanocidal, anti-malarial, anti-chagas, antifungal properties. This covers a wide range of medical conditions that can be fought using some of these synthesized quinoxalin derivatives. It is actually believed that these are more biologically active because of their N-oxide functional groups that are on the pyrazine ring of the quinoxalin structure. And so N-oxidation generally increases the biological properties of these quinoxalin structures. N-oxidation may also be essential for bioreduction and have been reported to specifically ease the reduction of the quinoxalin ring. Several studies have shown a relationship between the ease of reduction and their biological properties for certain groups of quinoxalin di-N oxides. And so this is why we are very interested in this group of structures. This is why I've chosen to study these structures and not other ones. And recently there was a series of Quinoxalino 2,3-C cinnolines and their derivatives and their 5N oxides that were synthesized, that after doing a thorough search through SciFinder, there was no evidence of any biological or electrochemical properties that have been studied for these series of quinoxalins. And so I'm interested to see what are their biological properties and what are their electrochemical properties, and can we compare these together and find any correlation to why their properties are this way because of their structure. And so I want to study this because redox reactions may play a role in their bioreduction, that may play a role in their biological properties. And so I would like to determine these to understand the mechanism of action of these specific chemicals, as well as to understand how we may be able to design some new drugs associated with the biological properties, and whether we can use these specific molecules to design drugs, or whether we can take these compounds and make them better to make and synthesize molecules that are related to these or derivatives of these that may be more effective in drug development. And so essentially the plan of action is to synthesize these desired 5N oxides and study them. And so most of these methods were actually for synthesis were taken from a journal published in 2011, and they in reaction 1, essentially we are going to take derivatives of benzofurazine oxide and derivatives of 2-nitryl nitrobenzyl cyanide, and react these two things together in acetonitrile. And we're going to add some pyrrolidine to this reaction, and essentially through this reaction, after about 12 hours of sitting at room temperature, we should have a solid, and then we will also have a product that is in solution. And so we are going to have to filter out the solid in order to use these for later to synthesize more desired molecules, which will be our 4A through E. But our 3 product will actually still be in solution. So we're going to actually have to perform column chromatography in order to get our 3 product out of solution. And so we're going to perform this, and we're going to store the 3 product for electrochemical analysis for later. But our 4 A through E products are going to be stored in order to perform more reactions in a multi-step synthesis. And so essentially we're going to perform this reaction 5 different times for each derivative. And then we're going to move on to reaction 2, which will synthesize our Quinoxalino 2,3-C cinnolin 5N oxides. And so this will, by reacting the stored 4A through E products, we are going to take each one of these products and we're going to dissolve it in KOH, and we are going to heat the solution for about 2 hours at 60 degrees Celsius, and there should be a solid that forms. This we will extract. We will purify it using silica gel column chromatography, and we will store these. We will perform this for each of the 4 derivatives, and we're going to store the products for electrochemical analysis as well. For reaction 3, we are going to do a benzylic acylation of methyl substituted to amino quinoxalines. And so essentially we are going to take that 4E product and we're going to perform that exact same procedure for reaction 2 on this product as well to create product 6. And then we are also going to do another reaction on this specific compound. We're going to dissolve it in tert-butyl alcohol in KOH solution, and also put some ethylene glycol in there. And the solution will be heated at about 80 degrees Celsius for about 30 minutes, and then we're going to cool that down. And then we're going to also use the silica gel chromatography in order to actually purify this. We're going to store this, both of these products as well for electrochemical analysis. So essentially we're going to perform electrochemical analysis on these compounds, these 7 products that were stored. And essentially we're going to do this through cyclic voltammetry. So essentially how cyclic voltammetry works is that it's a technique that measures the current that develops in the electrochemical cell under conditions where the voltage is actually in excess of that predicted by the Nernst equation. And this will be performed by cycling the potential of a working electrode and then measuring the resulting current. And so essentially it performs various cycles, and then it creates this graph that graphs the potential versus the current. And so essentially when we're looking at this graph, we are able to see what the reduction potential is, as well as how reversible the reduction actually is for this specific reaction. And so we are actually going to be using a 3 electrode cell, and how this works is we're going to have one of those electrodes is going to be a PT disc electrode, and then a PT wire auxiliary electrode, and then our reference electrode, as you can see in this diagram, will be AG and AgNO3. And so essentially we're going to deoxygenate it before actually passing it through the cyclic voltammeter. And then we're going to send these samples off to the biology department to study on the petri dishes the way that they react with specific bacteria, with specific cells in the body, so that we can understand their biological properties to actually do a comparison. So what we would expect from this study is that with an electron withdrawing group on the quinoxalin, that there would be a less negative reduction potential, and that this would make the bioreduction of the structure able to occur just more easily, which would essentially increase the biological activity of the quinoxalin. There's also been a recent unpublished computational study that found no correlation at all. So it is also possible that we could not find a correlation. Essentially, I believe that this is a very safe procedure because we will be using chemicals that really only require standard personal protective equipment, such as goggles, lab coats, and gloves that will be worn at all times to prevent any contact with the skin or eyes at all parts of the lab. And then we'll also be working in fume hoods to avoid any inhalation of chemicals. These are pretty standard. The only chemicals that really require extra precaution is that in reaction 1, the toxicological properties of 2-nitrobenzyl cyanide and 4-methyl 2-nitrobenzyl cyanide still are not entirely known. So it's possible that the metabolism could break these down and release cyanide, which can cause dizziness, unconsciousness, and even death. And so we'll take extra precautions not to ingest or inhale any of these compounds when working with them. And then pyrrolidine is also highly flammable, harmful, and corrosive, so we want to keep these in an environment that are far from any ignition. They will be stored, used, and disposed far from any ignition so that they are not dangerous to us. These are really the only safety considerations that need to be taken into account. We will be following all the chemistry department's safety guidelines by using our personal protective equipment and disposing of all chemicals in the proper bottles. And so this is very safe, does not require a lot of extra safety precautions. I believe that we have all the necessary equipment actually in the chemistry department. Most of these chemicals and glassware actually are in the chemistry department stock room. The only special equipment we really use are the CHI instrument, voltammetric and analyzer, which is actually available at the chemistry department. Extra resources that I have is that I actually have 2 experts in this field of study. Dr. Philip Crawford is an expert in cyclic voltammetry. He has done many electrochemical studies on quinoxalin derivatives, so he has extensive knowledge in this area of study. Dr. Kelly Gottlieb, she actually helped write the journal that first synthesized these derivatives, and so she has performed these successfully. So I believe that with these 2 experts, I will be able to successfully perform this experiment. This is a relatively inexpensive experiment. It is most of these chemicals are actually found in the chemical stock room, and so really just these 1 derivatives as well as the 2 derivatives are the ones that will be the most expensive. They're still relatively inexpensive with only a budget of $274. which for chemistry really isn't too bad. This is a experiment that can be performed in a relatively short time span. I can perform this experiment if I was working every day, 5 days a week, for 4 weeks straight, just a couple hours a day, to synthesize these as well as perform the cyclic voltammetry. And so essentially the longest part of this would be sending it to the biology department for them to do the biological property studies on the petri dishes, because you have to wait for bacteria to grow. So this may take a little bit longer, but with I would expect for this to take about a semester to be able to get the data back from the biology department and also be able to analyze this and write up a report. So essentially this is not a slow process, and this can be done in a relatively short amount of time with very impactful results. So in conclusion, this research is significant because it will expand our knowledge of the electrochemical and biological properties of these newly synthesized quinoxalin derivatives. Quinoxalin and dioxides have displayed a broad range of biological properties, and so we would expect that these might as well. Through studying their relationship between the electrochemical behavior and the biological activity of these new quinoxalin derivatives, we will be able to study their mechanism of action to understand how they may work inside the human body, to be able to develop new drugs to fight some of these conditions that are displayed through the biological properties. This can be done relatively safely, can be done in a relatively short time span, and relatively inexpensive. With the resources that I have at Southeast Missouri State University and the expertise here, I believe that I can perform this experiment successfully and have an impactful result from this experiment.